everyone. This is a hopefully pretty short video to address the special senses, which is chapter 15 in your textbook. So the special senses are going to include your sense of smell, which is olfaction, your sense of taste, which we call gustation. It's going to include vision, and then also equilibrium and hearing. So equilibrium is like your balance, your ability to sense where you are relative to gravity, essentially. And all of these special senses result because there is a stimulus that is detected at a sensory receptor cell. And that sensory receptor cell might be a neuron, or it might be some other kind of specialized receptor cell that in turn communicates with a sensory neuron. So either way, we're detecting stimuli from the environment, and it's being communicated pretty directly to the central nervous system and specific locations in the brain. Although it's a good idea to point out right now that everything goes through the thalamus. Just a reminder that the thalamus in the diencephalon is an important relay center, and pretty much all sensory information is going to go through the thalamus prior to reaching its destination in the brain. So let's start by talking about olfaction. First and foremost, olfaction's stimulus is going to be odors, which would be the colloquial term, but the sort of official term for odors is volatile organic compound. That's basically anything that off-gasses. So you have molecules in the air, and that's what we're detecting as scent or odor, because those molecules are able to bind to receptors inside our nasal cavity. And those receptors are actually the dendrites of specialized neurons. You have a lot of them, somewhere between 10 and 100 million, which is less than a dog, but not a whole lot less, actually. But dogs dedicate more of their brain to interpreting smell than we do, which is probably why they are so much more sensitive to smell than we are. Although we do also have a sensitivity to smell. Smell actually has a very strong association with memories for humans, and smell can elicit very powerful memories for a lot of people. So if we look over at this diagram over here, we can see here we are inside the nasal cavity. Those are the concha of the nasal cavity that we learned way back at the beginning of the semester. And you can see the cribriform plate of the ethmoid bone right here. And remember, the cribriform plate is the one that has all the little holes in it. And those holes are so that these neuron processes can go from inside the nasal cavity and pass through the cribriform plate to reach the olfactory bulb. And so the pathway for smell is that, right, when you breathe in these VOCs, or volatile organic compounds, those molecules will bind to receptors on the dendrites of these specialized neurons. And then if the binding of that molecule generates an action potential, that will be carried up to the olfactory bulb. So we start at the olfactory receptor cell, and then that signal will pass through the cribriform plate right here of the ethmoid bone, enter the olfactory bulb, and then travel along the olfactory tract. And ultimately, it's going to reach the olfactory cortex. But before it even gets there, it's going to go to the limbic system and the hypothalamus. And you might remember that the limbic system plays an important role in memory. So that might be the connection that we see between smell and memories. Okay, so that brings us to gustation or taste. I think most people know that you have several primary tastes: sweet, salty, sour, and bitter. You also have something called umami that might be new for some of you. That's more like a savory flavor. And your taste buds that can detect those tastes are located on several different kinds of lingual papilla. So lingual papilla are just epithelial projections that are on your tongue. They look kind of bumpy when you look at your tongue. You have valate, foliate, and fungiform papilla, and all of them contain taste buds, and they could contain any of those sweet, salty, sour, bitter, or umami. Sometimes they have a mix of them. But there is a fourth type of lingual papilla called the filiform, and these little projections do not have taste buds. So they're sort of the exception to the rule. So if we look at the pathway here, you can see that we have taste receptors on the tongue. And to get to the brain, those signals, those taste signals, are going to travel via 3 potential cranial nerves. So that's kind of interesting. The anterior, say 2/3 of the tongue, is actually innervated by the facial nerve, which is cranial nerve 7. And then if we move back here, we have the glossopharyngeal cranial nerve, which is cranial nerve 9. And then way at the back of the tongue is innervated by the vagus nerve or cranial nerve 10. So if we were to use like an eyedropper or something to drop sweet or salty or something, particularly flavorful onto someone's tongue in different locations, and they could taste it in some places but not in others, we could probably isolate which nerve might be damaged based on where on the tongue we place something that someone couldn't taste. So that's a little bit interesting. All of those cranial nerves, though, that are carrying signals about taste, they all go to the same place in the medulla oblongata, which is called the solitary nucleus. And from here, of course, it's going to go up through the medial lemniscus, and ultimately it's going to end up in the insula. And remember, the insula is that part of the brain that you have to reflect back the temporal and frontal lobe, and then you can see the insula underneath. So that's the gustatory cortex for where you interpret taste and connect it to previous experiences. And then in this corner over here, if I just blow this up a little bit, you can see the different shapes of the papilla, and you can kind of see the different locations where taste buds might be located. And then, of course, again, the filiform does not have taste buds on it. Now, for equilibrium and hearing, let's just remind ourselves of a couple of basics before we dig into any additional details. Equilibrium is all about balance, and the part of the ear that is connected to equilibrium are the semicircular canals, and there are 3 of them. They are these big loop-de-loop things I'll show you in the next slide. Inside of them are what are called otoliths, also known as ear stones, and they actually move around depending on the position of your head, and their location in the semicircular canal lets your brain understand the position of your head relative to gravity. So that's super helpful for understanding what's up and what's down. Equilibrium is all about the vestibular nerve, which is a branch of cranial nerve 8. Another branch of cranial nerve 8 is the cochlear nerve, and the cochlear nerve is all about hearing. So that's a big difference between equilibrium and hearing. They share the same cranial nerve, which is cranial nerve 8, but they are each a different branch. And again, equilibrium is all about the semicircular canals, whereas hearing is definitely about some other structures inside the ear. So when sound waves enter through the external auditory meatus, they will reach the tympanic membrane or your eardrum, and the vibrations of those sound waves will move the ossicles. Those are the stapes, and the anvil and the hammer. And then that's going to tap on the oval window, which will transfer those vibrations into the cochlear duct. And then they will travel through the cochlear duct and ultimately reach the organ of Corti, which is where there are these very specialized hair cells that are sensitive to vibration, and the hair cells can bend in response to those sound waves. And again, just as a reminder, hearing is the cochlear nerve branch of cranial nerve 8. So let's take a peek at some of the structures. So here's our external ear. This is the auricle or pinna, which is what you would just call your ear. And then we can see the external auditory or acoustic meatus, which leads to the tympanic membrane. And then on the other side of the tympanic membrane, we have the middle ear, and we have those auditory ossicles here. So you can see the stirrup and the anvil and the hammer in here. Those are the 3 auditory ossicles. That stirrup is going to be tapping right here on the oval window, and that will send those sound waves into the cochlea. And inside the cochlea, which kind of looks like a snail shell, is where the organ of Corti will be, and the cells there are receptors that can actually respond and generate signals in response to sound. But if we come back to the ossicles right here, and we go superior from the stirrup and the oval window, that is where you can see those semicircular canals. And those are involved in equilibrium. So as you bend your head or lean over or lean back, the position of those canals will change, and the little otoliths inside of them will fall and land in a different place as you move your head inside those semicircular canals. And then there are cells that can sense those otoliths. And so that's how your brain understands what's up and what's down. So again, this is going to be the outer ear. We're going to have the middle ear, and then we'll have the inner ear. Going back to the middle ear, I just want to point out the auditory tube. Don't confuse the auditory tube with the external acoustic meatus. The auditory tube is going to connect into the nasopharynx, which is what you might sort of think of as your throat, or kind of at the back of your mouth. And so that means that there is a tube that connects the middle ear to your throat, and it can act as a passageway or a conduit for pathogens. They can kind of climb their way up through the auditory tube, and they may arrive in the middle ear and find themselves thinking, this is a wonderfully warm, dark, moist place. I think I'll live here. And then middle ear canal infections would lead to a buildup of fluid, which would push on the tympanic membrane and cause pain. You also can get blockages, like if the lining of the auditory tube is inflamed, then it can be hard for air to move back and forth. And so you can get that pressure feeling inside your ears, and you're trying to swallow or yawn to open up that auditory tube in order to allow it to equilibrate, for the pressure to equilibrate. So there's a model in the A&P1 lab that showcases a lot of these structures really well in 3 dimensions. So that might help you to better understand them and how they relate to one another. Okay, that brings us to the eye. Let's start with the accessory structures of the eye, which is going to include the eyelids, eyelashes, the conjunctiva. This should be an A right here, which is just the superficial epithelium of the eye, and then the lacrimal apparatus, which is what produces tears. So if you look over here at this image, I'm just going to blow it up a little bit if I can. Doesn't want to move down for me, unfortunately. Okay, so we can see here is the actual lacrimal gland, which is where tears are going to be produced. And there are little tear ducts that link the gland to the surface of the eye. So as tears are made, they drain out the ducts and into your eye. And then they will move to the inside corner, where they will leave the eye via the lacrimal canaliculi, and they will travel down the nasal lacrimal duct and ultimately enter the nasal cavity, which is why when you cry, your nose runs, because the tears that don't pour out over your eyelids are going to go down the nasolacrimal duct and come out your nose. And of course, as they do, they're going to pull any mucus that's lining your nose with them, so it can make you extra snotty as a delight. Also, I just wanted to point out that pink eye, which many people have experienced, is really just an inflammation of the conjunctiva. And that can be due to an infection or allergies. But if it is an infection, it can spread very easily. So you really want to try to avoid touching your eyes. You don't want to introduce germs to your eye, and you don't want to share any germs that you have in your eye with anyone else. So we want to avoid touching your eyes whenever possible. Okay, so in lab, you're going to learn the basic anatomy of the eye, and these pictures do a pretty good job of showcasing some of that. So let's just remember the pathway of light is going to be that it's going to pass through the cornea, which is the clear part of your eye. It will enter the anterior chamber. It will pass through the pupil, and then it will pass through the lens into the posterior chamber where the vitreous humor is, and ultimately it will strike the retina. And if this area of the retina where the optic nerve exits is actually your blind spot, but there's another spot not visible in this diagram called the fovea, which is where you have your greatest density of cone cells that are sensitive to light. And so your best vision is on the fovea or macula. On the outside of the eye, we have the sclera, which is continuous with the cornea at the front. Underneath that, we'll have our retinoid pigment epithelium, which is a melanin heavy layer that helps absorb light that's entering the eye. So instead of it bouncing all around inside your eyeball, instead it gets absorbed by that RPE. So that's really helpful for giving us good vision. And then over here, we're just looking at it from a slightly different perspective. We're kind of cut down through the middle here. We can really see the ciliary body, which is where our aqueous humor is going to be produced, which is what fills the anterior chamber. And we can see our little suspensory ligaments, which support the lens and help adjust the lens. They can make it thicker or thinner, depending on whether you're trying to see near or far. And then you can also see the iris and how it circles around to form the pupil in the middle. And of course, the iris can change size to make your pupil bigger to let in more light when it's dark, and it can constrict to let in less light when it's really bright. So the pathway of light, just hitting this one more time, when light enters your eye, it'll pass through the cornea, it will enter the anterior chamber with the aqueous humor, and then it will pass through the pupil, through the lens, into the posterior cavity where you have the vitreous humor, which is more jelly-like, and then it will hit the retina. But the retina is actually a ton of layers. Okay, it's about 10. But the cells that sense light are all the way at the back. So the rod and cone cells are the deepest layer, which means the light has to pass through all these other layers before it finally reaches the cones and rods that can generate a signal in response to the light. So in this image here, you can see the cone cell, which lets you see color. You can see the rod cells, which let you see in the dark. It's more of a grayish green. And again, as I mentioned earlier, the macula or fovea is where you have the densest number of cone cells. So you have really good high contrast color vision on the macula or fovea, whereas your peripheral vision is more rod cells, which are more sensitive to light. They don't need the light to be as bright to generate a signal, which is why they let you see when the light isn't good, but then the contrast isn't as great, so there's a cost to it. So when light comes through the eye, it's going to pass through these layers of the retina, and then it will reach the rod and cone cells at the back, and then if those rod and cone cells respond to the light, then they will send a message first to the bipolar cells. And then from the bipolar cells, it will go to the retinal ganglion cells. And then the retinal ganglion cells will actually, their axons will form the optic nerve that you can see they run across the surface of the retina right here. And they will exit the retina at the optic nerve. And at that spot, because there's all those axons, there's no rods and cones. So that's why that's your blind spot. And then from the optic nerve, the information will travel to the thalamus, and the thalamus will send that information on to the visual cortex, which remember is in the occipital lobe. So just some interesting little facts about vision. The images in your eye are actually upside down and left, right, reversed on your retina because when light passes through the cornea and the lens, it refracts or bends. So the top ends up at the bottom and the bottom ends up at the top. But you don't even notice because your visual cortex actually flips them right side up and left, right correctly based on information coming from your semicircular canals, for example. So based on where your head is versus gravity, that's how your brain knows what should be up and what should be down despite the way the information is coming in from the retina. So you just fix it and you don't even notice. And then also myopia is when you're nearsighted, so you can see things near but not far. Hyperopia is when you are farsighted, so you can see things far away, but you need glasses for reading. And both of them can typically be corrected using lenses that just alter how the light is refracted so that it focuses on the macula just perfectly. Because essentially what happens with myopia and hyperopia is that either usually it's the shape of your cornea is either bending the light too much or too little. And so it's either focusing just in front of your retina or behind your retina, but not on your retina. And we want it to come to that perfect focus point right on the retina. That's how you get great vision. And that's what we're trying to do with glasses or contact lenses. Another option for correcting your vision is LASIK or PRK. PRK is the original version of LASIK. Basically, both of them use a laser to reshape your cornea so that it bends the light correctly. The difference is that in PRK, the laser is going to ablate the surface of your cornea. So it's just going to go right ahead and reshape the outer surface of your cornea. That means that it will use up less of your cornea in order to reshape it, but it also means it's going to hit a lot of nerve endings, and so the recovery time is a little longer and a little more painful. With LASIKs, they're going to laser a flap, and then they'll lift the flap, and then they'll use the laser to reshape the cornea underneath the flap, and then they drop the flap back down when they're done. So essentially there's many fewer nerve endings that are damaged by the laser, so it hurts a little less and the recovery is a little quicker. But LASIK also uses up more of your cornea. So if your corneas are thin to begin with, LASIK may not be the best procedure for you. Both PRK and LASIK have been used a lot over the last 20 years by millions of people with really great outcomes. But there are also complications and negatives that come with both procedures. You may end up getting a really bad glare effect at night or have double vision. So, again, all surgeries come with risk, and that's something that you have to weigh as a patient. All right, so that is our short lecture on the special senses, and you can read more about them if you like in chapter 15 of your book.